Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
Inside every single one of your cells, right now, there's a vast library of instructions being read. That library, your DNA, holds the blueprint for, well, everything that is you. But how does a cell actually read that blueprint to build and maintain a living thing? It all starts with a truly beautiful process called transcription. So let's dive into this molecular marvel. So here's our game plan. We're going to start with the big picture, the absolute fundamental flow of genetic information. Then we'll get to know our key player, RNA, and see what makes it different from DNA. After that, we'll walk step by step through the main event, transcription itself, from how it starts to how the message is built, and finally how it knows exactly when to stop. You know, to really understand transcription, we've got to start with the foundational rulebook of molecular biology. This is what's known as the central dogma, and it basically describes how information gets used inside a cell.
And this diagram just lays it out perfectly. Think of your DNA as the master blueprint. It's priceless, so it's kept safe inside the cell's nucleus. You don't want to risk damaging it. So, to use the instructions, the cell makes a temporary, disposable copy. That process, that's transcription. The copy, an RNA molecule, then travels out to tell the cell how to build proteins in a process called translation. So, DNA to RNA to protein. That's the core of it all.
Okay, so if DNA is the master blueprint, what exactly is this copy? Let's get to know the star of our show, ribonucleic acid, or just RNA. It's the versatile messenger molecule that actually carries out DNA's orders. So at its heart, RNA is a chain of building blocks called nucleotides, which sounds a lot like DNA, right? But here's the key difference you can see here. RNA is typically single-stranded, and that one simple change in its shape has a massive impact on what it can do, making it this incredibly flexible, adaptable tool for the cell. And the differences don't stop at just one strand. This table breaks down a few more crucial distinctions. For one, the sugar in RNA is ribose, not deoxyribose. And famously, where DNA uses the base thymine, or T, RNA swaps in a different base called uracil, or U. Finally, RNA is way less stable than DNA. And you might think that's a bad thing, but it's actually a feature. It's a temporary message, so you want it to break down easily after its job is done.
Now, RNA isn't just one thing. It's really a whole family of molecules, and each one has a very specific job. The one we're focusing on is messenger RNA, or mRNA. That's the one carrying the code to build a protein. But you've also got ribosomal RNA, which actually builds the protein factories, and transfer RNA, which brings the building materials, and there are many others too.
And check this out, because it really puts things in perspective. Even though we talk about messenger RNA all the time, it's only about 5% of the total RNA in a cell. The vast majority, a whopping 80%, is ribosomal RNA. It's the literal bricks and mortar of the cell's protein factories, so it needs to be everywhere.
All right, so we've met our messenger molecule, RNA. Let's get to the main event, how it's actually made. This is the transcription process, where that genetic code is carefully and precisely copied from the DNA blueprint.
I love this analogy, I really do. It just perfectly captures what's going on. The cell doesn't photocopy the entire DNA library every time it needs something. No way. It finds the one specific gene, the one page in the book it needs at that moment, and makes a copy of just that one instruction. It's incredibly efficient.
And the molecular machine that's responsible for this amazing task is an enzyme called RNA polymerase. Think of it like a highly skilled, incredibly fast scribe. It zips along the DNA, reads the sequence of letters, and perfectly writes them down into a new RNA molecule. But this whole process doesn't just start at some random point on the DNA. It's very, very precise. The first critical step is figuring out exactly where to begin. This stage is called initiation.
It all starts with a specific sequence on the DNA called a promoter. You can think of this promoter like a big sign that says, "Start copying here." The RNA polymerase enzyme spots this sign and binds on tight. As soon as it's locked in, it unwinds a small section of the DNA double helix, exposing one of the strands so it can be read.
And this image shows that moment perfectly. You can see the RNA polymerase enzyme, or RNAP for short, latched onto the DNA right at that promoter region. The start site is marked, the DNA strands are peeled apart, and we're ready for the next phase. The machine is primed and ready to go.
Okay, so with the process initiated, the RNA polymerase now gets down to its main job, actually building the RNA message, one nucleotide at a time. This stage is called elongation. So during elongation, the polymerase just starts cruising along the DNA template strand. As it moves, it reads each DNA base it passes, A, T, C, or G, and grabs the matching RNA nucleotide to add to the chain. And remember that rule. Where it reads an A on the DNA, it adds a U to the RNA. It does this at an incredible speed, adding around 40 nucleotides every single second.
And this is what it looks like in motion. As the RNA polymerase chugs along the DNA track, this new RNA transcript just peels away from the template behind it. And the DNA that's already been copied, it just zips right back up into its double helix. It's this beautiful, continuous, dynamic process.
So this polymerase is just meticulously building the RNA chain, sometimes for thousands of letters, with amazing accuracy. But genes have a beginning and an end. So how does this little molecular machine know when it's reached the last word on the page? How does it know the message is complete?
Well, that brings us to the final act in our story, termination. Just like there are start signals built into the DNA, there are also stop signals that tell the RNA polymerase, okay, your job here is done.
And the stop sign isn't just a simple period at the end of a sentence. It's a special sequence of nucleotides right there in the DNA code. One of the most common signals is a G-C-rich region that's also a palindrome, which just means the sequence reads the same forwards and backwards on opposite strands. When this gets copied into RNA, it triggers the end.
So here's how that trick works. When that special palindromic sequence is copied into the new RNA strand, the RNA itself actually folds back and sticks to itself. It forms this tight little hairpin structure called a stem and loop. This structure physically bumps into the polymerase, which causes it to stall and just fall right off the DNA. And just like that, the newly made RNA molecule is set free. And this diagram just gives us that perfect bird's eye view of the whole journey, start to finish. You can see the RNA polymerase locking on at the start, moving along the DNA during elongation, and then finally detaching at the end, releasing that finished RNA transcript. From a simple signal on DNA to a functional messenger ready for its next mission.
So transcription is complete. We now have a messenger RNA molecule that is a faithful copy of a genetic instruction. It holds the code. But how is that code, which is written in the language of nucleotides, how is it actually read and translated to build a complex three-dimensional protein? Well, that is the next incredible chapter in this story.